Thanks, Bob, and the organizers for giving me the opportunity to talk today. It's great to see some old friends in the audience. So I don't know if Bob remembers this picture. We went together to a conference, food exhibition actually. It was in Milan, Italy, and it was an amazing meeting we had with Bob. And I saw this quote from Ed Wilson about biodiversity and the complexity of understanding and studying biodiversity. So basically, you need to really take sort of small pieces of diversity, either in a habitat or community, and or taxonomically looking at only a small portion of what is out there. And then I have this other quote that is also from Ed Wilson from his later years that says that basically biodiversity in one sense is everything living. And I think that we are in at a time right now that we can actually get eDNA to address the challenge of studying biodiversity in a more holistic way. So if we look at the tree of life, we'll see quite a lot of diversity, but then in order to, again, be able to functionally address this from biodiversity tracking, we have looked at different compartments, like invasive species, pests, species with, for food and health related products, or bioindicators, ecological indicators, and so on, and some of the endangered species. So again, one thing that I always argue is that we should not forget about this axis of time and space. This idea of being able to track biodiversity is not a static thing. I'm done with my PhD thesis. I've studied my group of organisms, and that's it. No, we have to repeatedly measure biodiversity and also cross space. That kind of makes the challenge bigger. Now, I'm going to jump quickly into societal implications to some extent, to reality. Look at the ocean sector. We'll see some traditional sort of fields. I don't know if you see my cursor here. Yeah. So you will see, for example, we have this traditional sort of industries, transportation, offshore oil and gas, fisheries, and so on. And we have a number of emerging, I would say, industries. Like the big one is this renewable energies like offshore winds or even deep sea mining. And all of these require understanding how we're going to impact biodiversity. And ocean is a very challenging environment. In the past few years, I've had the privilege of working with some of the groups that explore ocean biodiversity, and I've appreciated more the difficulty in this case. So we are living in at a time that biodiversity is now sort of being highlighted. Let's say we talk about biodiversity crisis or biodiversity decline, but we also need sustainable development. We need more resources to deal with the challenges that we have. These are some of the 5 threats to biodiversity that sort of been highlighted by United Nations. I'm not going to go into that, but land and sea use, pollution, species overexploitation, climate change, and invasive species. And throughout the conferences of eDNA, almost all of these are highlighted as applications, or potential applications, shall we say. This is a global map of all, in just the meta-barcoding studies that have been published since 2020 or so, that we compiled in a review article. And you will see that these studies are from all over the globe. But the challenge is that most of them are done in a framework that is not directly applicable to those industrial and societal implications. Most of these studies have been focused on artisanal, smaller scale, proof of concept, or looking at discovering biodiversity and also optimizing protocols and so on. When we look at the On the other hand, like, of course, we'll see regulatory groups, regulatory agencies, and some of the NGOs are starting to utilize eDNA, but we have some important shortcomings and missing pieces. I give you an example from an industrial side of things that I have worked with more. This is from the oil and gas and energy sector. A few years ago, we basically worked with their scientists, and they actually were good enough to form an eDNA or environmental genomics research program within their basically the environmental association that we have. And as you can see here, there are a number of applications that they've highlighted within the, let's say, the life cycle of a typical project. A lot of them are same type of things that we discussed: invasive species, endangered species, population level analysis, measuring abundance, biomonitoring, and so on. But one of the things that these groups highlight is in order for them to adopt and utilize eDNA technologies, they require standards. And these standards, like, basically are important for a number of reasons. Is that when you try to change, let's say, the focus of, let's say, an environmental sort of assessment project towards a new technology, and then you want to apply it globally for all of your assets, then obviously you want to enforce it in a standardized fashion in a way that it can be cost effective, feasible, and so on. These are actually from their website. This is public information. So you see this is the basic request for proposals. And they so far have requested 4 or so. Most of them, almost all of them, have links to establishing standards, conventions, best practices, and so on. And that's where I think that our community needs to be more proactive. And I'm glad to see that conferences have panel discussions and so on. I highlighted some of the challenges. For example, the primers. We still see quite a number of different primer types used, or markers, or fragments of markers. We saw in Chris's talk, for example, different fragments, different library coverages for those fragments, and so on. These are some of the examples that we have worked on. There is the evolving sequencing technology. That's another challenge. Like, you adopt a method using a particular sequencing technology and let's say read length. And then here comes the new technologies, and there are new errors, new kind of base colors and so on. And another big challenge is the data analysis side and bioinformatics, where this is a new article that just came out, and we contributed to, and there are 33 or 31 or so pipelines available for bioinformatics analysis of metabarcoding data. So let's say from an industrial sort of user, which one do I use? Which one is better than the other? And so we need to have some framework to basically deal with these uncertainties. And this is good stuff, but we also need to consider some of the challenges. So during the pandemic, I thought about this in a more measured way, so I had a lot of time in my basement at home, and thought about this notion of how do we validate and standardize, eDNA approaches, and what are the tools we have, what are the frameworks we have. So we do have some serious challenges, and we saw that in a presentation about the abundance, today. That, like, what are we proving here? Are we trying to detect just the molecules in the water or in the environment? Or is the goal, which I think that's our goal, is to infer the presence, absence, and perhaps abundance of organisms using those molecules as proxy, which I think that these are different challenges, and one is easy, the other one is not so easy. We need to consider that as well. So how do we use the tools that we have, or the frameworks that we have? And I'm not going to go into this in details, but I highlighted this, and this is published in an article in Trends in Ecology and Evolution, to sort of 4 different categories for validation, from validation approaches that we have: experimental validation, comparative and ecological validation, bioinformatics validation, and statistical validation. I'm not going to go through it, but each of them have their own pros and cons, and we need to consider that an absolute truth in this case is difficult to reach, so we have to be very careful about the validation and how we use it. Another way that we can in fact validate or recognize the use of eDNA is when it's used in the legal framework, and this is the famous Asian carp issue in Great Lakes, and in other contexts, when you look at other technologies, when they start to be utilized within a legal framework, a number of checks and balances have been met, and it's been recognized. And in this case, we can learn quite a lot from the forensic DNA, and they have established excellent frameworks for the use of DNA evidence within different jurisdictions, and then eDNA practitioners can learn quite a lot. There are some differences between eDNA and forensic DNA, but there is a lot of similarities. The other thing that I would say that is really important, and it would be great to see more of it, is the validation through official statements. And we saw that, for example, Chris talking about what's happening with US Congress and mandate. We've seen some of the work from DFO in Canada. We've seen some of the work between, let's say, NOAA and then groups in Europe, for example. These official statements, white papers, practical guides, and so on, they all help position eDNA for the applications, and again, that sort of brings more, de-risks the use of it, shall we say. But on the other hand, we also need to recognize what do we mean by when we say we are using eDNA, and is it good enough or not? Is eDNA simply a modern replacement of what we used to do with traditional methods? I argue most of the cases are leaning towards that. They have been like chasing fish using, nets or observing whales, using binoculars or acoustics, and now they want to use eDNA for it. I would say that we need to go beyond that. We need to go beyond that, especially as we're dealing with the biodiversity crisis and climate emergency. So that's where I think that we need to think about like a little bit outside the box and then put eDNA to the good use. And that we have learned a great deal from the COVID, where we know that technologies should not be like 100% bulletproof before they can be used, because we're dealing with the urgency of the issue, and this is a comic about the wasp, which is relevant in fact to the eDNA. So in that context, a few years ago, we established a standalone center linked to a private company, and we, I'm not going to go through it, but what I say is that by doing this and bringing a number of partnerships that you can see here, and dedicated team, you can achieve quite a lot, and I'm quite happy and proud with their achievements. They have done over 200 projects with partners, more than 200 environmental samples, and 5 million species detections, and about 9 trillion bases of DNA sequences. And this is all a dedicated center which have gone through a standardization, in this case with ISO, and establish the workflows from sampling all the way to reporting within the ISO framework. And I get into that in a minute, and I know I'm running out of time very quickly. So the other way that we can basically use eDNA in a societal sort of side is through the involvement of indigenous and community groups. And I was quite skeptical about this myself. I thought, oh, we cannot do this DNA sampling, like using someone who never touched a, pipette or doesn't know how to sort of do these type of sort of DNA-friendly methods. But I was proven wrong with my friends and colleagues in some of the NGOs that we work with, like Living Lakes. And it started this project called STREAM, which is focused on macroinvertebrates, and now we're doing also diatoms across Canada. And this is through partnerships and modularizing the project and the standardization on top of that, where we do have different modules of the projects done with different groups, and it's been coordinated here at the university in my lab. And we have done quite a lot of work. I'm not going to go into detail, but there's a 15 seconds. This actually happened about 2 weeks ago in Ontario. And they all get their certificates, you know, so they do get the standard training and certificates, and we have about 300 people right now across Canada that are certified to do this type of sampling and contributing either for our projects or for any other type of eDNA based analysis. And this is our data portal. It's a work of Artine, a master student in my lab that put the portal together almost a summer, and we have different levels of sort of analysis that you can see here, and a bunch of publications. The other thing is that we have been working very closely with our agencies. This is the work we're doing with the CABIN, which is the Canadian Aquatic Biomonitoring Network, led by Donald Baird, the science led by Donald Baird, and we're comparing the method that we have with the traditional approach here. And as you can see here, we have better resolution through the DNA approach. You've seen these results in other contexts, but this is, again, the data that is generated through community work can actually help the regulatory side, not just for outreach. It's actually going to generate data that can be used for biomonitoring. And the same thing, we can ask different questions from this data, and this is the last sort of couple of slides I have. This is using that data to get more insights. My postdoc, Andrew, has been looking at the data that we're getting in the stream and finding phylogeographic and population level patterns. And again, we have two different amplicons. They nicely match. They look at and then we do statistics on it, and we see that they actually corroborate quite well. So these are not pseudogenes or noise in the data. Finally, I think this is sort of a new kind of group that some of you guys here might have heard of, that we put together is for eDNA community, because I have been seeing a lot of national or regional standardization happening, but nothing truly international. So we are bringing together representatives of different networks, different initiatives, like DNA Aquanet, the GEDWIG, the US Marine workshop that Chris mentioned. And we are forming this, we have formed this international eDNA standardization task force so that we can fast track the development of the international standards together with the work that we are doing with ISO, and to see these standards really established in a timely fashion. So if you're interested, please reach out, and happy to chat about it more. And time for lunch.